Welcome back to NalaTalk, where we discuss updates on the field of genetics testing, especially its implementation in clinical settings. As usual, my name is Fadhli. I'm the product lead and pharmacist in Nalagenetics, and we have together with me Dr. Esther, the doctor, and also the research lead in Nalagenetics. How are you, Esther?
Hi, I'm very good. Another week,
great.
Another NalaTalk.
Another week, another NalaTalk. Cool. Yeah, so
what
now
are we going to talk about today?
Yeah, I was thinking like now that we're nearing the end of the year, where we've been talking about the diseases that comes from feasting, like gout and hypertension. Maybe it's a good time to talk about do a little bit of recapping of the knowledge that we've been learning about. So maybe we talk about like pharmacogenomics, basic and fundamental. What do you think, Esther?
Yeah, absolutely. No, you'll be my guide, actually. I'll throw the questions to you. But yeah, I think that's a very fascinating topic because just a little bit of a background, I think some people think that once you graduate from med school, like you know everything that there is about medicine, about diseases, and also about genetics. But in fact, like if you look at all the syllabus, there's so many things that we have to learn in such a short amount of time. So genetics or pharmacogenomics, more specifically, is actually covered very minimally. And I think when I started working at Nalagenetics, I discovered all these things that I didn't know about genetics and the implications in the clinical settings, as what we always said. So I was just hoping to share more about that for our listeners out there who might be interested. Maybe we can start with some of the different terms that are used, because I know that people sometimes use it interchangeably, even though they mean different things. For example, just to start with like pharmacogenomics, and then there's pharmacodynamics, and then there's pharmacokinetics. So as a pharmacist, like this is really something that was honed in maybe in school, isn't it? Yeah. So can you elaborate a little bit about the differences?
Right. So that's a cool question. Like every time I get that question, like so let's break it down, right? So the umbrella topic for all of this is actually called pharmacogenetics or genomics. So pharma means whatever it's related to the drugs or the compounds that is usually used to treat a disease inside our body, while the genetics or genomics means in relation to the gene or the genome. So initially, there's a difference between pharmacogenetics and pharmacogenomics. Genetics means that you only look at a particular gene as the thing that will affect the how the drug is affects in your body, while genomics is something where you look at it as a whole, as a collection of genes, because genome is actually the whole collection of genes. But right now, as the field progresses, people usually use it quite interchangeably when you talk pharmacogenomics and pharmacogenetics. So under the field of pharmacogenetics, there's actually two ways. One is pharmacokinetics, another one is pharmacodynamics. So that's a whole mouthful, right? So we just look at the difference here is kinetics versus dynamics. Yeah, so pharmacokinetics is the way that our body reacts to the foreign substances, and that is including the drugs. So on the other hand, pharmacodynamics is the way that the drugs affect the body. So on pharmacokinetics, we look at how the body reacts and try to expel the drug, while the pharmacodynamics is how the drug actually go to the target and then exact its intended purpose. So there, in pharmacokinetics as well in pharmacodynamics, in both of them, genes are actually involved, because most of the way we respond to the drugs and get affected to the drug is through enzymes or other kinds of proteins. And these proteins are coded by the genes that we have. So in the mutations in the gene will actually affect change in the enzymes or the protein. So yeah, that's basically the short form of what is the difference between these three terms.
Yeah, no, it's all coming back to me, and with like all the diagrams and the form of distribution and such, it's just like all the terms. I was like, oh yeah, it brings back memories. And you mentioned mutations, like in the protein or in the enzymes. Maybe can you expand a little bit more about how do we detect the mutation? I mean, we talk about like pharmacogenomic tests. What is actually involved? Like, what is specifically that we are looking for? And what are some of the different types of sequencing, different methods of sequencing that we have right now?
Yeah. So before we go into the way of how things are mutated, how we detect mutation, right? Maybe we can talk about like the different types of mutation that's present inside our DNA first. So why is there mutation in the first place? It's because our cell is constantly growing. It's constantly replicating in a process called like mitosis. And during this kind of replication, there sometimes can be an error in terms of replicating, because as filler or as accurate as our cell wants to be, sometimes the checking mechanism can be a bit erroneous. So a replication process involves a step where the DNA gets rewritten into another part so that it can become two cells. So during this kind of rewriting or duplication, there can be an error, and this error can come in different ways. One is when there's a missing letter inside. So instead of you replicating like 4,000 base pairs, you only replicate like 3,999. So you lose out one. Or there can be an extra letter added or extra base pair. So instead of 4,000, it becomes 4,001. So this kind of mutation is usually fatal, and it does not support life, because if you don't know, the genes are actually read 3 base pairs at a time. So if you remove one base pair, there can be something that's called frame shift mutation. So everything doesn't make sense. So if you imagine like a novel, right? If you struck out the word N from A and B, it becomes add, and then you add another letter from the other sentence, and it becomes like gibberish. So it doesn't make sense. So this kind of frame shift mutation is not the one that we want to look at. Something that is more, what do you call it, insidious, or something that you won't be able to see right away, is when there's polymorphism, or there's a switching of the base pair. So instead of like written like cat, C-A-T, it becomes like hat. The C is replaced with a H. So this frame still is going to be read as per normal. There's no shift. But on that base pair, there's a change in the kind of amino acid that's being used to build that protein. So that is what we want to find out. So knowing that we want to find out that little changes inside our genes, like that, those letter changes, we can actually do several ways in the way of detection. So if you imagine the whole genome as a novel, one way is you can do a Control F, right? You can Control F, and then you try to find out a word inside, like cat. So you know that the word what you want to find out, you put in cat, and then there's actually like 75 hits. So you expect to have a 76 hits, but now you have 75 hits. So that's a way. And this method is called PCR, or polymerase chain reaction. So you look for a specific sequence that you know it should be like this, but if it doesn't look like that, it will be a mutation. Another way is actually you can do direct sequencing. So direct sequencing is when you read the whole text from start to finish. So this takes this is also called usually Sanger sequencing, or a more modern version is called next generation sequencing, where you read the whole thing, and it's actually more accurate, but it's more resource intensive and also time consuming. So that is another way to find out. So there are things like in between as well, where you look at different things. Also, a difference in not only the way you look at it, the region that you look at also has different ways. So there are methods where you look at only the whole exome. So whole exome sequencing, because what you know is that the whole genome, right? Not all of them are supposed to make sense. Only about a percentage of them actually have the gene that codes for the functional protein. Some of them are just like rubbish that you don't know what it does, or it's just
we don't yet know.
Just to preserve the whole gene structure. So we don't want to read that. Like for example, there's the poly A tail that where on the end of the gene where it will actually just to preserve the whole content of the gene. So yeah, we can do whole exome sequencing, or we can read the whole genome itself. So maybe that's a good.
That's very great. I was like, oh my God, it's just like sitting in a lecture. No, but I'm glad that you expand a little bit more about that, not just mutation, but the different types. And also, like as you explained it, I could picture like the diagrams, the animation that we used to be shown in order to explain it. It is very complex, and there's a lot of intricacies that are involved. But it's definitely fascinating, and I'm sure people out there are interested to know as well, and there'll be heaps of development in the future in this field as well. So I think you mentioned about the change in base pairs. Would this be called the single nucleotide polymorphism that we normally refer to? And can you expand a little bit more about that, the differences and how it contributes to function?
Right. So yeah, great. That's a great question. So we usually call the changes in the base pair SNPs, single nucleotide polymorphism, or SNPs. Usually, what causes a change in terms of the function of an enzyme doesn't come from just one point of mutation or one SNP. It usually comes from a collection of SNPs. And also to remember that the DNA is like that you have two sets of DNA, one from your father and one from your mother. So usually you look at both of them, and usually a mutation on one strand could actually be neutralized by a normal strand from the other strand, a normal sequence from the other strand. So yeah, so when we look at SNPs, right, we look at when we want to see the polymorphism, right, we want to look at more than one SNPs. So a collection of more than one SNPs itself on one strand can be called a haplotype. Before that, the different categories or different permutations of SNPs are called alleles. So usually you have the wild type allele or the reference allele, and then there's also the mutant allele that actually the one that is being changed. So a collection of alleles will be seen as a haplotype, and then if you put it together, both from the father and the mother, a set of two haplotypes will be a diplotype. And usually, when we look at it from the clinical perspective, we look at the diplotype that gives rise to the characteristics that we actually see in the person. So, for example, I have like 10 SNPs that you want to take a look, and these 10 SNPs will give rise to the haplotype that I have, and I have like 5 mutations on that 10 SNPs, and then on another SNP I have another 5 mutations, and you put it together, that what make my color of my eyes black, or the color of my hair black, something like that. Yeah.
I think people's head are started spinning already.
Yeah.
No, but just to add to that, actually, what about variants? Because we hear about that a lot. Like, how is that different to what you explained before?
Yeah. So variant usually is something that you assign to in terms of in reference to the reference, right? So a reference allele will have different kinds of variants. So you have like, for example, a reference of like CYP2C9*1 will have like a variant of like CYP2C9*2 and *3. So just a variant is called, a variant is what is being compared to from the reference, is what I'm saying. Yeah.
I mean, these are very brief introduction. I mean, we can go for hours, and people actually have to go take courses, in this, but I think it's just important for us to just touch upon so that people know there is a whole new world of genetics to be discovered. And I think you mentioned earlier about the differences in the sequencing methods, like one being kind of a PCR and the other would be a more either whole genome or exome sequencing. And those usually, the two different types also use different machines, right? Yeah. And that also affect the result that we can get, like in terms of the accuracy as well as the time and the effort that it will take as well. Is that right? Yeah.
Absolutely right.
And also, I think earlier you mentioned that we don't only look at the difference in the SNPs or in the variants, but also there are other things that can influence the activity of the drugs in the body, such as the pharmacokinetics and pharmacodynamics. And for example, like, can you give an example, for example, like receptor expression and other things that have a role to play as well?
Yeah. So the way that a drug is actually processed in the body is like multifactorial. So the gene polymorphism is basically just a single one factor of it. So what it dictates is it dictates the shape of that enzyme, which will actually conform to whether the drug is going to be easily linked to the enzyme and get metabolized easier, or exert the effects much faster or slower. But it doesn't actually talk about the expression of this gene. What do you mean by expression? What I mean is that how much of these protein will be created by the body. So nobody actually is talking about that. So we only talk, when we talk about pharmacogenetics, right, we only look at the conformation of that. Conformation means the shape of that protein. So we don't know yet how much of this protein is going to be produced inside the body. It could be like 100 or 200 or 1,000 or less. Mm-hmm. And the way and this actually affects the metabolism as well as the effect of the drug itself, because the drug expression will actually cause to whether the drug is going to be metabolized faster or slower. And of course, these are actually affected by many things like age, the way you eat, your lifestyle, your gender, and even like what other drugs are you eating at the same time. Like, for example, we can take an example of the drug like ibuprofen, or the painkiller drug. Painkiller drug could be affected by CYP2C9, like it is actually metabolized by that. And if you actually take other drugs that are that occupies the space of CYP2C9, like for example, like warfarin, you can actually reduce the metabolism of ibuprofen. And then also for patients who are still young, the CYP2C9 expression might be still lower, so you can have overdosage of ibuprofen, and many others as well, examples.
Now, talking about multifactorial things that can affect the gene expression, with Nutrigenetics, right? It actually usually looks at a number of different genes that can sort of interact with nutrition. Can you explain a little bit more about Nutrigenetics? Because I think that more and more people are becoming aware of this test. But what is actually the purpose, or what do we actually look at when we're talking about Nutrigenetics?
Yeah, so that's interesting, right? Because nutrition compared to like pharmacogenetics is something that our body is used to have. So our body produces these kinds of like vitamins in ourselves, and we do need it for like healthy sustenance. So when that happens, our body already is accustomed to these kind of molecules. We usually have not only one pathway to follow in order for its processing as well as its absorption. We have multiple. So when there's multiple pathways, there will be a lot of like fail-proof mechanisms where where if one pathway fails, there's going to be another way for us to get it. So when we talk about the field of nutrigenomics, we don't talk about a single gene effect. We'll talk about multi-gene effect. And usually when we talk about multi-gene effect, we can't use just the simple method of like qPCR, where you only look for a certain SNPs or a certain mutation, because it's going to be expensive because you need to look a lot of things. What we do is we're going to have to do like a microarray or like a next generation sequencing, where we look at multiple genetic factors and impute it together to get the results. So this is quite interesting because in nutrigenetics we look at the effect of a number of genes giving just a little effect on the mutation or on the metabolism of the nutrition, and then actually it can give up the results. We can't only look at one gene, because if we look at one gene, there might not be any effect on it. Yeah, that's interesting.
Yeah, well, I thought I'll just touch on it because you mentioned like multi-genes and multifactorial things that can contribute to it. But I think like we can expand more about nutrigenetics in the next few episodes. But just to sort of recap, we've talked, in the prior weeks about the use cases of pharmacogenomics, and today I hope that everyone here can learn more about the technical aspect of things. Maybe some terms were a bit foreign to you, and today you can understand what we mean by that. And obviously, there are a lot of things that are left to be discovered out there in the field of genetics and genomics. And maybe you can be one of the pioneers. Who knows? But tune in to our next episode where we talk about nutrigenetics a little bit more with experts. Until then, take care and see you.